if we have a cell in the body, right, let's say this is the cell membrane, this is the nucleus with some DNA here, right? And then we have the RAF endoplasmic reticulum here with some ribosomes on it, right? Good. And then we also have some mitochondria here and there, right? Some tiny vacuoles, some fluid in it that we call cytoplasm. Remember that? Good. So there is a human cell, right? An animal cell. And then let's say here we have a blood capillary surrounding or moving close to that cell, right? Good. Now, you see blood carries substances towards the cells, right? The blood supplies our cells in the body with substances like what? Like water, right? So there is water going into the cells. Let me put it here, H2O. There is also oxygen going into the cells, transported by blood. Let's say O2, that is oxygen. And there is also food, right? Nutrients. Let's say here C6H12O6, that's the formula for glucose, right? So we say H2O stands for water. O2 is oxygen, and C6H12O6, that is glucose. These are three substances that are constantly being transported by blood towards all cells in the body, right? And there is a next one that we need to mention here, which is carbon dioxide. But you know, carbon dioxide moves in the opposite direction. It comes out of the mitochondria where it is produced, and it leaves the cells and goes into the blood, right? That is CO2. So CO2 stands for carbon dioxide, right? Well, on top of that, there are other nutrients like amino acids, right? Vitamins, minerals, that are also transported by blood to the cells, right? To be used for cellular activities, right? Like for example here, there is a lot of sodium ions in the blood and in the fluid. By the way, there is a fluid surrounding the cells. We draw here around the cell, right? A fluid that is called what? What do we call that fluid around the cells? We call that tissue
So
fluid.
that would be tissue fluid.
We can call it tissue fluid. You can also call it extracellular fluid, right? And it's very important because that fluid has roughly the same composition as what we have in the cells, right? So substances that leave the blood go into the tissue fluid first, and from there they go into the cells, right? I mean, they don't go straight from blood into the cells. They need to go into the surrounding tissue fluid first, because you see, cells in our body, guys, are kind of floating in that fluid, right? Yes. When you have a burn on the skin and you see kind of a fluid coming out of your skin, a water-looking fluid coming out of the burn. I don't know if you have ever seen that, or if you remove accidentally part of the skin, right? Let's say you scratch it, you fall down and scratch your skin. You may see kind of a water-looking fluid coming out. Well, that is tissue fluid, meaning that cells in your body are surrounded by tissue fluid. And they are also surrounded by blood capillaries, as you can see here on this diagram, right? Blood capillaries. And these blood capillaries supply the cells with these substances here. Right, guys? Now, how do these substances go in and out of the cell? Well, they can do it by different methods, right? So let's say we call that transport. We call this movement of substances transport of substances. And substances can be transported by passive transport or active transport. What do we call passive transport? There are two methods. One is diffusion and one is osmosis, right? Or osmosis. And active transport, we just call it like that, right? Active transport. Now, what's the difference? Passive transport happens spontaneously, right? Does not require energy, right? Let's say occurs, how do you spell this word? Like this, spontaneously. Maybe you can help me out with this. And that means without any
Yes,
help,
sir.
right? It happens spontaneously. That means it happens on its own with no help, right? Spontaneously, and does not, right? Doesn't require energy from the cell, right? Does not require energy from the cell. However, this one, active, means that uh, requires energy, right? This one requires energy. Why is it that this one does not require energy and happens spontaneously, and this one, however, does require energy and needs a push, right? But these two guys, these two ways of moving substances in and out of the cell happen spontaneously and don't require energy because substances are moving from a region where there is a lot of them into a region where there is fewer quantity or none. Meaning that, for example, look, oxygen is transported from the lungs. Just imagine the millions of red blood cells that we have in the blood. Those red blood cells, they all carry oxygen. You see, all these red blood cells in your blood carry oxygen. Meaning that there is a greater quantity, there is a greater concentration, a greater amount of oxygen all the time in your blood compared to in your cells. And then oxygen will be moving from a higher concentration into a lower concentration, right? I always give an analogy here. Look, let's say this is a bus or a train, right? That transports people from one station to another, right? when the bus or the train is full of people, right? And they need to get out of the bus, well, that will be easy because outside there is more space, right? And then they will be just moving from an area of greater concentration where there are many people, right, into an area of lower concentration because this is the station here, right? So there is a lot of space and people will be scattering around, right? It's easier to move. And it's easy to move, right? It's easy to move when you are coming out of the bus or out of the train. Actually, you will feel relieved. If the train wagon was very full of people and you are coming out, see, you will do it spontaneously. You will be doing it, right, spontaneously. You don't need any help, right? You will be moving from an area of higher concentration into a lower concentration, just like oxygen does from the blood into your cells, just like glucose does, because there is more glucose in your blood than in your cells. This glucose is coming from the intestines, right, from the small intestines. The oxygen is coming from the lungs. Water is also coming from the small intestines. So in the case of these substances here, they will be moving from a higher concentration into a lower concentration. When it is oxygen and glucose, we call the process diffusion or simple diffusion, right? Diffusion. And when it is water, just for the particular case of water, when water moves, right, from where there is more water into where there is less water, we call that osmosis, right? So look, diffusion is the movement of, maybe if we want to come up with a definition here, we can say that diffusion is the spontaneous movement, right, of particles from a higher into a lower concentration region. Right? We can also say here that the substances will be moving in favor of the concentration gradient, right? In favor of the concentration gradient means from higher concentration into lower concentration. Guys? Now, if it is water, it's the same thing. Water molecules will be moving from an area where there is a higher concentration of water or more water into where there is less water, right? So this is a particular case of diffusion. We can say this is the diffusion of water, right? Diffusion of water. Now, one difference is that diffusion may occur outside the human body in different situations, and it does not need a membrane actually. I mean, all the time. You see this cell membrane here? For diffusion to take place, right? If we talk about the human body, well, yes, substances will be diffusing through the cell membrane. Like this carbon dioxide here, you know why it moves in the opposite direction by diffusion? Because look, it is being made in the mitochondria. It is a gas. So there is a higher concentration in the cell, and then carbon dioxide will be moving from a higher concentration into the blood where there is less carbon dioxide, where there is a lower concentration. But that will be diffusion also, right? Now, I was saying that diffusion, guys, may also take place outside the human body. Like, for example, here. You see this slide here? Okay, what we see here on the right-hand side is a glass of water that we have put, let's say, a food coloring in it, right? This blue food coloring, you put a drop in the water, and then the particles will be moving from a higher concentration, right? From a higher concentration into a lower concentration, and the particles will be scattering. Eventually, the whole water will turn blue. Ain't it? Well, that is a particular case of diffusion. That is diffusion. That is an example of diffusion. But there will be diffusion in liquids, for example. But here there is no membrane. We are not talking about particles diffusing through a membrane like the cell membrane, right? This is a gas. Look, bromine vapor. In this container, it has a glass lid on it. If we take a similar container and put it upside down on top of this one, right, and remove the glass lid, right, in such a way that the gas can move upwards, we will see this eventually, right? Kind of half of the gas would have diffused, right, upwards from a higher concentration at the bottom into a lower concentration on top. And that will eventually make the concentration even. Actually, that is the purpose of diffusion, to even the concentration, right? Particles will scatter and scatter and scatter around until the concentration is the same all over. Guys? I just brought these examples here because diffusion may take place every day around you, and probably you didn't know that that is called diffusion. Like, for example, you are in your living room and someone is cooking in the kitchen and something is burning, and then you start smelling that burnt food in the living room, right? Or in the dining room. And how come? If there is no wind blowing that smell towards you, well, that is actually because particles will be scattering from the kitchen and will be moving from that higher concentration into a lower concentration all over the house. Eventually, that will come towards your nose. We call that diffusion. See what I mean, guys? Diffusion is around you every day. However, if we talk about diffusion in the human body, there are some examples that you need to know about, right? Like this one here, right? The diffusion of glucose from blood into your cells, the diffusion of oxygen from blood into your cells, and the diffusion of carbon dioxide from cells into your blood. Because this carbon dioxide now will be taken away from the cells and will be transported back to the lungs so that we can breathe it out. I mean, the blood will go back to the lungs so that we can breathe out the carbon dioxide. Now, in the lungs, something very interesting happens also, because let's say this is your lung here, right? Your left lung, your right lung, and then let's say this is the trachea, right? The larynx, and eventually you have your nose here somewhere, right? Your nose. Right. And then oxygen, you see, is taken in when you breathe air, right? When you inhale air, a lot of oxygen will go to your lungs. And then that oxygen will be taken to some tiny balloons that we have in the lungs that we call alveoli, right? So let me take one of these, let me zoom one of these bunches of alveoli. It will look like this. One there, one there, one there, one there, right? A lot of alveoli. And then the oxygen comes in, right? The air, I mean, these tiny balloons will be filled with air, right? And then they are surrounded by blood capillaries, of course. You know what happens? Oxygen diffuses from the alveoli into the surrounding blood capillaries, and that makes the blood oxygenated. Then that oxygenated blood, right, will now go from the alveoli to the heart, right, to your heart, and from your heart to your body. And that blood will take oxygen to your cells. See how interesting this is? Right, so cells will be surrounded by blood capillaries and oxygen will diffuse from the blood into the cells. So there we have the diffusion of oxygen. Right? So oxygen diffuses twice. In the lungs, it diffuses from the alveoli into your blood, right? And then the blood will transport it to all cells in the body, right? When it reaches the tissues, the oxygen will come out of the blood into your cells. So there we have two examples of diffusion of oxygen. Oxygen diffuses from the alveoli into your blood, and later on it diffuses out of the blood into your cells. Guys, glucose diffuses, right, from the intestines, from the small intestines, glucose diffuses into your blood. And then the blood will transport it to your cells. When it reaches the cells, it will diffuse into the cells. See that? So glucose also diffuses twice. From the intestines after digestion, it diffuses into your blood because here there are blood capillaries surrounding, right, the intestines. Right? So a lot of glucose, every time we eat, a lot of glucose will go into this blood by diffusion mostly, right? And then later on, the glucose will diffuse out of the blood into your cells. Right, guys? And the other example of diffusion in the human body is carbon dioxide, for example, which is produced in the cells, will diffuse out of the cells into your blood. And then the blood will take all that carbon dioxide, will go back to the heart, and from the heart, it will be sent to the lungs to release the carbon dioxide into the alveoli. So here we have blood capillaries. Let's draw them in blue because that blood is not oxygenated. And this one in blue carries CO2 from the body. Right? CO2 from the body. See, from all cells in the body, CO2 goes to the lungs and diffuses out of blood into the alveoli so that we can breathe it out. Right? So here will go, CO2 will go out of the blood into the alveoli and then will come out, right? CO2 will come out here, right? Up the trachea, and then we breathe it out. We breathe it out, CO2. So where is the oxygen going to? It's going to your cells. Where is the carbon dioxide coming from? It's coming from your cells and transported by blood. And for that to happen, we need diffusion of both gases. In the alveoli, let me write it here, alveoli, right? This is in the lungs, right? Lungs alveoli. In the alveoli, there is something that we call gaseous exchange. Right, guys? Gaseous exchange. That means that oxygen and CO2 are exchanged. Oxygen goes from the alveoli into your blood and carbon dioxide comes out of the blood into the alveoli. That's why we call it gaseous exchange, right? And in your cells, there is also diffusion when oxygen and glucose leave the blood and carbon dioxide enters the blood by diffusion. Right, guys? Well, in plants there is also diffusion because in the leaves, for example, we will see later on that for photosynthesis, plants will need carbon dioxide to go into the leaves. And that is done from the air where there is more carbon dioxide than inside the leaves. So carbon dioxide will be moving by diffusion into the leaves. At the same time, oxygen is produced during photosynthesis and oxygen comes out of the leaves and will be coming also by diffusion, right? Because there will be more oxygen than in that portion surrounding the leaf surface, right? But those are examples of diffusion in plants also. And I would say that's an example of gaseous exchange also in plants, right? Now, what about osmosis? Osmosis is the diffusion of water. Remember, look, diffusion of water. But there is one great difference, and that is that osmosis requires a cell membrane. It requires a material for the water to move through, right? We can do it in a lab, but we will need to find a material that is suitable for that, right? Like for example, look, in the lab, we may use one kind of a bag, right? That is made up of a kind of a plastic bag, but it's kind of porous. Porous. I mean, it has pores, kind of tiny pores, tiny, 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 very tiny pores. That material is called a whisking tube. Right? A whisking tube. You can buy it. We can buy it, right? And then what we can do with this whisking tube, guys, is let's say we can put water in it and some sugar. Right? Let's say some sugar and some water in it. sugar water, right? Sugar water, kind of sweet. Remember, it has very, very tiny pores. Now what we are going to do to demonstrate how osmosis takes place, guys, is that we are going to put that Viskin tube into a beaker with some pure water in it. Right? Pure water. So this water outside is just water. Right? Actually, we don't call it pure water. We call it distilled water. Distilled water, that is water obtained by distillation. You take the water, boil it, and then you condense the vapor. You get distilled water, which is pure water, right? Pure water. And then look at what we have here. We have water in the beaker, and we also have water in the Viskin tube. But the one in the Viskin tube is not pure water. It has sugar in it. It is a solution, right? Now, eventually, we are going to see something happening if we are clever enough and we put here, let me say, a tube, a capillary tube. That is a very thin tube, right, inside the Viskin tube. We are going to see at the beginning some of the sugar water goes up, right? Some of this water goes up. Let me draw here with a yellow line, right? Look, guys, look inside this capillary tube. A capillary tube is a very narrow tubing, a very narrow tube, right? So look, I'm gonna draw some inside here, right? Inside here, just look at what is going on inside here, right? I'm going to draw some of the sugar water moving up. As soon as we put the tubing, look, right? Some of the sugar water will be moving up and up and up to a certain point. Let me say up to here, right? Up to here. If we take a close look at the tubing, right? Zoom, probably this is what you will see, right? There, and then you have the sugar water went up to this point, right? So there you have the sugar water. See that? Up to that point. And that line, that curved line there on top, right? We are going to call that the meniscus. The meniscus is actually the top line of a liquid in a container. If you have water in a container, the surface will not be flat. It will be kind of bending a little bit inside there, right? So like this water inside this beaker, the surface will be kind of this, right? Kind of bending a little bit upwards by the edges. You see that? So it will be kind of this, right? In a liquid in a container, the surface will be kind of bending upwards a little bit. And we call that line the meniscus. So look at where the meniscus is, right at the beginning, huh? Right at the beginning. So here we have sugar water. I should have used the yellow color. Sugar water. We have pure water outside. Now guess what? What do you think will happen 2 hours after? Think. Considering that the Viskin tube is porous, very tiny pores. One, one possibility, the water outside will turn sweet, meaning that some sugar came out of the whisking tube into the surrounding water, making the surrounding water not pure anymore, but kind of sweet a little bit. That is one option. And then the level of this water will rise, and the level of inside the whisking tube will decrease. That could be one thing to consider that may have happened. And the other thing, guys, is that water from the beaker, this is a beaker. Right? This container is called a beaker. So water from the beaker would have entered the whisking tube. And then the level of water would have decreased here, and the level of water in the whisking tube would have increased, and then the meniscus would have moved upwards and probably would have overflown this capillary tube. What do you think may have happened? Well, to answer this question, we need to know about osmosis. So the whisking tube, this material that I said is porous, right, is actually what we call a partially permeable, partially permeable, partially permeable membrane. Can be considered as a partially permeable membrane. We can also say semi-permeable. Right? Partially permeable, we can also say semi-permeable. So semi-permeable means partially permeable, which means it's not totally permeable, which in turn means that this material, guys, will allow only very tiny molecules to move through and not big molecules like sugar. So in this particular experiment, the only molecules that will be allowed to move through this whisking tube, since it is a partially permeable membrane, will be water molecules, not sugar molecules. Look at the formula of sugar. Huh? The formula of sugar is C12H22O11. However, that of water is H2O, meaning that one molecule of water has only 3 atoms. One molecule of sugar has 22 plus 12 plus 11, all this bunch of atoms in one single molecule. Right? That's why here we can say that this whisking tube, this semi-permeable membrane, will allow only water molecules to move through it, not sugar molecules. Right? So this is banned, not allowed. Water, yes, will be allowed. Now the question here is, in which direction will the water molecules move at a greater rate? Actually, they will be moving in both directions, right? But they will do it at a faster rate in one direction. What direction will that be? Well, water molecules move through a semi-permeable membrane always in the direction in which there is less water on it, or it is more solute concentrated. So water will be moving from where it is watery into where it is thicker, you see, more concentrated. Therefore, in this particular case, since there is just water outside and there is water and sugar inside, water molecules will be moving at a greater rate, right, into the whisking tube. The sugar will not be allowed to leave the whisking tube. Sorry, I didn't tell you that before. Right? That's why you may have said that the water will be contaminated. Right? because I didn't say that the pores were not big enough to allow the sugar molecules to move through. Right, guys? Sorry about that. I didn't give you that piece of information before. So yes, this is what will happen here, guys, right? If we have this setup, water molecules will be moving into the Viskin tube. And therefore, 2 hours later, this level of water would have decreased, right? So let's say it's no longer there, right? Now it is somewhere here, let's say here, right? And then the level of sugar water in the Viskin tube would have increased, right? Its volume. The Viskin tube would have probably got fatty, kind of, right? And then this meniscus would have moved upwards because of the pressure of the water that is coming into the Viskin tube. Guys, this is a past paper question. Huh? Let's see this one first. So it says, state the meaning of each of the following terms, diffusion and osmosis. Well, we already saw that diffusion, this is the spontaneous movement of particles, right? Particles in favor of the concentration gradient. You can say in favor, you can say along the concentration gradient, right? You can also say from a region of higher concentration into a region of lower concentration, right? So this in favor of the concentration gradient, right? Let me put a straight line there. That means thus from a region of higher concentration to a lower concentration region. That is what we mean along the concentration gradient or in favor of the concentration gradient. However, osmosis, right? This is the movement, or we can say the diffusion, right? The movement of water molecules through, don't forget to mention the membrane, right? Through a partially permeable membrane, right? From a region of higher water concentration. This means lower solute concentration. Or you can say more dilute, right? Or you can say a dilute solution. dilute solution. Means from where it is watery, right? From a region of higher water concentration or a dilute solution into a region of lower water concentration. Which is a thicker solution, right? A concentrated solution. Concentrated solution. But that is defining osmosis in all its terms, right? Kind of a lengthy definition, right? Right. Item B, figure 1 and 2 represent experiments to show the movement of molecules. So here we have what? We have a beaker, some water, we put some solute, right? Let's say like a solid food coloring or dye. Eventually we see that the particles are moving here, guys. Look, from this region of higher concentration at the bottom, you see the particles will be scattering about, scattering, and eventually the whole water would have turned, right, into a homogeneous color, right? What process is that? What we call this process? Hello? The process of
Diffusion, sir.
particles. Huh?
Diffusion.
Diffusion, yes, this is diffusion. This here is diffusion. That's diffusion, right? Diffusion may happen of a liquid in a liquid, a solid in a liquid, right? Let's say a gas in a gas in a liquid, right? Provided that particles will be moving from a region of higher concentration into a lower concentration until the concentration is even, is homogeneous, right? All right. Look at this other experiment. This is the Viskin tube that I was talking about. This is the same setup, right? Concentrated sugar, solution, and water outside. Both experiments show the movement of molecules, suggest two differences between the movement of molecules in this experiment compared to this experiment. Well, two differences. One, in the first experiments, there is no membrane. No membrane for the molecules to move through. And here there is a membrane, right? There is a partially permeable membrane. The other difference is that the particles that diffuse here is a solid, right? So this is a solid solute, a solid solute diffuses, right, in water, let's say. And here is a water, right, diffuses. There is water on both sides, but the water will diffuse into where it is thicker, right? From where it is more watery or just water into where it is thicker, more concentrated, more solute concentrated. You see, guys, in this experiment, there are two ways of making the concentration on both sides even. One, if we bring some sugar into this water, if sugar could have come out of here into this water, eventually we would have had sugar solution in here and sugar solution out here, both of the same concentration, both of the same level of sweetness, right? However, sugar molecules are not allowed to go through the tubing. The other way is to make this solution in here taste the same as the water outside. And to do that, we need to make this one very watery, very dilute. And that is actually what happens. This water will go in there, trying to make this solution inside there as watery as possible. Right? It will always, it will never be the same concentration as outside because here we have just water. But what if we had, guys, a container with, let's say, a semi-permeable membrane, right? A semi-permeable membrane there, right? Semi-permeable membrane, and then we put some water on both sides. Let's say here we have side A and the other part is B. And then look at what we are going to do. Huh? We are going to put, let's say here we are going to put 1 teaspoon of sugar, right? 1 teaspoon of sugar. Like this, right? And then on the next one, we are going to put 2 teaspoons of sugar. 3. We make 3. 1, 2, 3. 1, 2, and 3. Right? Teaspoons of sugar. And then all this sugar will come into here, and there will be 3 times more sugar here on B than on A. Guys, are you following? Hello? Just talking and talking, but I
Yes,
hope
sir.
you are.
Yes, sir.
I hope you are watching my screen and
Yes, sir.
trying to figure out what is going to happen here, right? Number one, this is a semi-permeable membrane, a partially permeable membrane. Partially permeable membrane, meaning that only water molecules will be allowed to move through this membrane, not sugar molecules. How can diffusion of water take place, meaning which direction, in order to make both solutions taste the same sweetness? Let's say taste the same. Right now, this one is sweeter, isn't it? This one on B is sweeter, of course, right? It's 3 times as sweet as the one on A, on side A. But how can we make them taste the same? There are 2 options. If we could transfer, right? Let's say one teaspoon of sugar from this side to A, but they will taste the same, isn't it? But unfortunately, we cannot move the sugar. How can we make both taste the same if we cannot move the sugar?
We can diffuse water from A, so there will be less water in A.
Yes, exactly. I agree with you. If water moves from A to B, then A will become more concentrated because it will have less water in it. It will taste sweet. Huh? And this one that is getting water will taste watery. Eventually, both sides will taste the same. And that is exactly what happens in real life, guys. Water will always move from where it is watery into where it is more concentrated, in order to make the concentration on both sides the same. We call that process again, we call that osmosis, right? So osmosis is the movement of water molecules from where there is a higher water concentration. You can also say from where it is a dilute solution. So this is a dilute solution, right? And this one is a concentrated solution. So water will always move from dilute into more concentrated. And here we are talking about solute concentration, not water concentration, right? Because water concentration, there is a greater water concentration on side A, right? In each portion of this container here, A, right, there is more water than here on B, because here there is sugar, and wherever there is sugar, there is no water. When it is just water outside, well, that is clearer, that is easier to understand. But when we talk about two solutions, well, here we need to look at that, right? Water will move from where it is watery into where it is thick in order to even the concentration. In real life, there will be a moment here in which both sides will taste the same, because here the level of water would have decreased and it will be increasing on the other side. So this part A will taste sweeter and sweeter every time, whereas part B will taste watery eventually, right? And they will taste the same eventually. Now, this, guys, brings me to a next situation here. Let's say we have three containers, right? This comes in the exams also. What I'm going to explain now, guys, comes in the exams. So here we have one container, 2 and 3, right? And they all have some water in it. Water in it. Right? So there is water in there. Pure water. Right? Well, actually, what we are going to do is we are going to put some red blood cells here. So let's say we're going to put one red blood cell here, one red blood cell here, and one red blood cell here, right? Red blood cells don't have a nucleus, so we are going to draw it like this. They have a lot of this is one of the fewest animal cells that don't have a nucleus, right? What they have instead is a lot of hemoglobin molecules to transport oxygen, right? So these are red blood cells. Now, guys, the first red blood cell is in a pure water, right? What we have here is pure water. Remember, when we say pure water in the exams, they can also say distilled, right? Distilled water. That is pure water. The second scenario here, the cell is going to be in a salty solution. So here we are going to add some table salt to the water and make it salty, right? And then here we have a salt water. We call that salt water, right? Just like seawater. And here, in the last scenario, what we have is a solution of same concentration as in the red blood cell. So meaning that, guys, the concentration of this water outside here is the same as that inside the cell. Remember, this cell has also a cytoplasm, and there is water inside the cells. Our cells have water in it. So let's say the concentration of this solution is the same as that in the cell. The question is, what will happen, guys, eventually? Let's say 2 hours later. Would these cells have experienced any change? Would have them experienced any change? Okay, let's say in the first one, you have a cell, there is a solution inside the cell, there is a cell membrane, ain't it? There is a cell membrane here. You see, cells have a cell membrane. A cell membrane is a semi-permeable membrane. Cell membranes are semi-permeable membranes just like this one, or like this Viskin tube, right? You see, guys, this cell membrane, that's the cell membrane that is present in all our cells, right? Red blood cells, white blood cells, skin cells, muscle cells, they all have a membrane similar to this one. Right? And that membrane is semi-permeable. What do we mean by that? It will allow certain substances to go through, but not big molecules. Huh? Among these tiny particles that can go through is water. Water and some ions like sodium, potassium ions, right? So what will happen here, guys? What do you think will happen in the first case? We have pure water outside, we have a cell membrane, and we have a solution inside the cell that we call the cytoplasm, right? So what will happen? Will the water move into the cell, out of the cell, or will not move at all?
So I say it would move inside the cell.
Yes, it will move inside the cell. I agree with you totally, because you see, remember that osmosis is the movement of water from where it is watery, from where there is more water, isn't it? From where there is a greater concentration of water into where there is less concentration of water. So this cell will get water in it. And you know what? Will eventually burst. It will burst. So unfortunately, it will do this, right? Will burst. So let me put it here. The red blood cell, right, gets water by osmosis. This is what will happen eventually, right? The cell gets water by osmosis, and it got so full of water that eventually it bursts. It is destroyed. Right? So when this happens, guys, you have this scenario that it is more dilute or watery, more watery outside the cell than inside. We say that this medium is a hypotonic, right? Let's say cell, let's say red blood cell in a hypotonic medium. when it is more dilute or more watery or just water outside the cells, we say that that medium is hypotonic. Hypo means below normal. Now, however, what do you think will happen here, guys, on the second scenario? We put a red blood cell in salt water, and this red blood cell also, of course, has a cell membrane which is partially permeable. And this water outside is salty, meaning that this is more concentrated than inside the cell. Inside the cell is more dilute, kind of more watery than outside. So when you have this case that is more concentrated outside, in which direction will the water move? Into the cell or out of the cell? And in this particular case, you know what will happen? Water will be coming out of the cell into the surroundings. And this cell will become dehydrated. Right? So eventually, this cell will kind of shrink like this, right? This cell will kind of shrink because of losing water. And this is actually what may happen if you stay like kind of for too long in the sea. Have you heard that people become dehydrated? It's not only because of not drinking water, but also because of being inside the sea water, right? You may lose some water from your skin cells because it is more concentrated outside. See what I mean? So when it is more concentrated outside, we say that is a hyper a hypertonic medium, right? Red blood cell in a hyper means greater than normal, hypertonic medium. And this one here, guys, since the concentration of this water solution outside, maybe that is salt solution, is the same as that in the cytoplasm of the cell, you know what will happen here? Well, water will still move through the cell membrane, but it will do it at the same rate. Water goes in, water comes out. Goes in, comes out at the same rate, and then you don't see any difference, right? When the solution outside the cells is the same as that inside the cell, we say that that medium is isotonic. Isotonic solution. Right? So that is a red blood cell in a iso means the same, right? Isotonic solution. So as a summary, guys, if it is more watery outside the cells than inside, water will go into the cells, and the cells may burst because animal cells don't have a cell wall to protect it from bursting, right? If cells remain in a medium that is more concentrated, thicker, than inside the cells, then the cells will lose water by osmosis also. This one here is what happens when you sprinkle some table salt on cucumber slices, guys. You see, let's say you have a cucumber, right? And then you slice it. And when you slice it, let's say this is these are the seeds, maybe. Guys, if you if you sprinkle some table salt on it, have you noticed that when you sprinkle some table salt on cucumber, water comes out? Have you ever noticed that? I invite you to do that experiment at home next time you prepare a cucumber salad, guys. Just sprinkle some table salt granules, grains on the cucumber. You will see very quickly water forming around. It's kind of the cucumber slice starts sweating, kind of, right? You know why is that? That is because the table salt, when it touches the cucumber surface, which is wet, this table salt will dissolve and will form a very salty solution in that region. And because it's very salty, it is a hypertonic solution compared to inside the cells. And then the cells start losing water. And then water forms around there. I invite you to do that. That is osmosis in plants. Right? Again, why is it that on Charlotte Street, when you see the vendors, right, selling, let's say, tomatoes, right? Why is it that they sprinkle water on the vegetables, especially when it's very hot? Have you ever seen them spraying water with a bottle, probably? Water on the vegetables to make them look fresh, to stay fresh. You know why? Because of osmosis, because this water is just water. It will go into the cells, into the tomato skin cells, right? And will make the tomato uh, uh, hydrated, right? And will look fresh. And what about, guys, a plant cell that has a cell wall and a cell membrane? The cell membrane is partially permeable. The cell wall is permeable, totally permeable, right? So here we have the cell wall, which is permeable, and the cell membrane, which is partially permeable. Right? And then we put this, let's say this is an onion cell, right? You put it in a container with some water in it, just pure water, just pure water. Distilled water, right? So what I'm going to do is, since I'm going to use it 3 times, let me put this one here. This one here. So look, guys, what will happen? Let's just repeat the experiment that we did with the red blood cell. Let we do it with this onion cell, right? Which is a plant cell. So if this is pure water, right, or distilled water, on this other one, this is salt water, and this is just a solution of same concentration as in cell. Right? So what will happen? Same as before, right? You see, since the cell wall is permeable, water will go in. Here, salt will go in through the cell wall, and water also. Salt and water will go through the cell wall. Right? Whatever solution is here, so maybe here we can say salt solution, right? Salt solution, but not thicker than inside like here, right? It's the same concentration as inside the cell. So the cell wall will allow anything to go through, but not the cell membrane because the cell membrane is semi-permeable, right? Partially permeable. Here, since it's just water outside, and here in the cell, remember we have the cell sap, which is this in the large vacuole here is just water, and water with some substances dissolved. Remember we call that the cell sap, SAP, right? And that region, that structure that holds the cell sap is the large vacuole. And there is where the cell takes the water in. When water moves into the cell, it will go straight into the vacuole, right? Mostly. Though there is also water here in the cytoplasm, of course. Right? All right. Now, what happens here is, as before, water moves from outside, that is just water, into the cell, that is a solution, right? The cell does not have pure water in it. It's water with dissolved substances. So since we have pure water outside, that pure water will be moving into the cell by osmosis, right? And then the vacuole would enlarge, isn't it? It will take up more space, will now be full of water. So this cell is going to look like what? This cell that takes a lot of water in it. I'm dedicating a lot of time to this, guys, because this comes in the exams. Huh? As you saw at the beginning, some questions. So look, the cell membrane would be kind of pushed towards the cell wall, and then the nucleus too, right, will be kind of pushed, and then the vacuole would have enlarged like this, right? Compared to this one. You see that one? Look at what it will look like now. But you know one great difference is that this cell will not burst. Just like this one here did, the red blood cell when it takes water in eventually bursts, while here the cell, the plant cell, when it takes water in, it does not burst. Why is that so? That is because of the cell wall. The cell wall is tough and rigid and will prevent the cell from bursting. Right? What happens is that when the cell is full of water, it will not take more water in because of too much of pressure inside, right? So the water that tries to still get in will be pushed away by the high pressure inside the cell. Right? So here's what happens. The cell will not burst. It will still take water as much as it can, right? But will not burst. However, this other cell here in this other scenario, right? Remember, what happens here is that there is salt water outside. This is a hypertonic medium, right? Hypertonic medium or solution outside. The solution outside is hypertonic compared to inside the cell. This one here, since it's just water outside, this is a hypotonic, right, medium. So when it is more concentrated outside and more watery inside the cell, water will move out of the cell. Water will move out of the cell. And what will happen to that cell? That cell will look like this. The cell wall is rigid, it will not change, right? Because it is rigid. However, since the cell was losing water, the cell membrane would detach from the cell wall, right? Will move away from the cell wall, and the vacuole would have shrunk like that, right? Because of the cell losing water. You see that? When that happens, we say that the cell is plasmolyzed, right? Has become plasmolyzed, meaning dehydrated. It's full of water, plasmolyzed is dehydrated. What about here? If it is outside is the same concentration as that inside the cell, remember what happens here is that water goes in and out of the cell through the cell membrane at the same rate, and then the cell would have not experienced any change, right? Because the medium outside is isotonic compared to inside. Osmosis or osmosis in living things. Like here we are going to refer to humans and plants, right? But when it comes to osmosis, we know it is the movement of water. So every time water goes in and out of the cells, right, uh, that is osmosis. So you can put it that way, right? And um, when water goes into your cells, right, it does that by osmosis, right? If water comes out of the cell, it does that by osmosis. Let's say when you are dehydrated, guys, when you are dehydrated because you are not drinking enough water or none, right? You are not drinking any water. The blood becomes thicker, becomes more concentrated, more solute concentrated, right? Because it has less water in it, because you are not drinking water, right? You know what happens? Why you become dehydrated is because water starts coming out of your cells, coming out of your tissues into your blood, right? So in that case, water moves in the opposite direction here, right? And then your skin becomes dry, right? Your lips dry, your mouth starts to dry, your lips may start cracking eventually. You see your eyes also kind of dry, and those are signs of dehydration. Right? You stop urinating in order to save as much water as possible. If you pee, by any chance it will be little and very dark because you are trying to save water, right? So every time water moves into your cells or out of your cells, then it does that by osmosis, right? Plants also. Plants take water through the roots by osmosis, right? The roots have some cells that are called root hair cells, right? There is where the water goes into, right? And then those cells eventually will be moving the water, right, from cell to cell, because when this cell here takes water and it gets a lot of water, there will be more water on this one than on the next one. So water will move from this cell into this one, and from this one into this one, and from this one into this one. You see, and eventually goes up the roots and up the stem. Right? So one example of osmosis in humans and animals is when water goes into your cells or out of the cells. Right? When plants take water up the stem through the roots, it does that by osmosis. Right, guys? The absorption of water in the intestines, when you drink water, the water goes into your blood. And it does that by osmosis. Right? So here we have some notes for you to read, guys. So look, the importance of diffusion in living organisms. I already explained this one, the diffusion of oxygen and glucose and carbon dioxide. So I'm going to leave this on the video for you to read over later on, right? And here is the importance of osmosis, right? This is the definition, and then examples of how water moves, right, in living organisms. Water moves into animal cells from blood plasma, right, and body fluids by osmosis. This keeps the cells hydrated. That is what I just explained here. Here, water moves, right, from blood into your cells normally, or from blood into the surrounding tissue that we call tissue fluid, and it also has water in it. You see? So water will move from your blood. Let's say you drink water in the intestines, in your intestines, right, guys? Remember, they are surrounded by blood capillaries also, right? So water will go from the intestines into your blood, and it does that by osmosis. And then the blood will transport that water to your cells, right? So here the water will be moving by diffusion, but we don't call it diffusion of water, we call it osmosis. From blood, where it is watery, right, compared to inside the cell, into the cells. You see this tissue fluid here is watery, it has water in it. Inside the cell is also watery because it's the cytoplasm. It has water in it. But the solution in the cytoplasm is always the same concentration, or almost always, normally, same concentration as outside. So outside is isotonic. It's an isotonic medium compared to inside the cell. And then the water will be moving out of the cell and into the cell at the same rate. The same as in your blood, Water will come out of the blood into the tissue fluid, and from the tissue fluid into your blood at the same rate. Unless you stop drinking water. If you stop drinking water, then the blood will become thicker, right? And that is very dangerous. And then what we do is we put water from the body into the blood, right? Where is that water coming from? It will be coming from the tissue fluid and from the cells, right? So when the tissue fluid donates water to the blood, it becomes thicker, and then the cell donates water to the tissue fluid. So in any case, the water will be moving out of the cell into the tissue fluid and from the tissue fluid into your blood. That is when you are not drinking water. But in general, you can say that. You can say that water goes in and out of the cells by osmosis. Substances can also go in and out of the cells, guys, through the cell membrane by active transport. And this one differs compared to passive transport like diffusion and osmosis in the sense that these two occur spontaneously and they don't require energy. Right? Active transport. Okay, look, active transport. Say we have a cell, right? This is the nucleus, some DNA, endoplasmic reticulum here with some ribosomes, some mitochondria here and there, some tiny vacuoles. Right. And then we have the cytoplasm, which is the jelly-like solution inside the cells, right? Water and substances dissolved. Right? Now look, we have inside the cell, let's say, sodium ions, Na, a little amount of them. Let me put 3 of them, Na. We have a lot of sodium outside, Na. These are positive ions, Na. And we have a greater concentration of potassium ions inside the cells. Let me put many of them, right? However, we have fewer on the surroundings of the cell. Remember, we call that tissue fluid, right? On the tissue fluid outside here, right? There is a greater amount of sodium ions and there are fewer potassium ions, right? However, these ions are normally moving in and out of the cell depending on the cell requirements, right? Now, when the sodium needs to be taken into the cell, that will be easy because there is a lot outside and they will be moving by diffusion from a higher concentration outside into a lower concentration inside, ain't it? They will be moving spontaneously, no help required, right? No energy involved. What if the cell needs to get rid of some sodium ions? Look, there are fewer inside, and there are many outside, plenty outside. And the cell still needs to get rid of some sodium ions to bring some potassium in. Well, that will be difficult, right? It will be like remember the train wagon I mentioned before, or a bus, and it was full of people inside, and then the train reaches the station, right? What if instead of these people coming out, more people wanting to get into the train? More people want to get into the train. Just like in Japan, Have you heard, guys, that in Japan, in the metro station, there are workers who are paid for pushing people into the train? Because the train reaches there already very full, and there are people waiting, needing to go to work, and they still try to push and push themselves into the train. And then there are helpers. There are people who are paid. In every train station, you see the metro station, you know what I'm talking about? There are people who are paid for pushing others into the train. So that requires a lot of energy because these people need to be pushed because the train is already full. You see, guys, that is what we call here in the cell active transport. These sodium ions are fewer, lower concentration inside the cell, a higher concentration outside the cell. And if the cell needs to take some of them out, that will require energy from the cell because that requires a push. That will require a protein here, a protein on the membrane that will open a channel for the sodium ions to move out. So these three things, it requires a membrane, it requires a protein, a membrane protein, and that will require energy from the cell. What is the energy in the cell? You see, energy which is obtained through respiration in the mitochondria. Remember, part of that energy is stored in the cell in some molecules that we are going to call ATP molecules. So every time sodium needs to come out, it will be moving from a lower concentration into a higher concentration against the concentration gradient. That will require at least two things. One, energy for the push, and two, a protein that will open a channel to facilitate that transport, transporting protein on the membrane. Guys, which is different from diffusion. If sodium was moving on the opposite direction, it will be doing it from a higher concentration into a lower concentration, and energy will not be required. At the same time that sodium comes out, potassium may move in, right? But potassium will also be moving against the concentration gradient because there is more potassium inside the cell than outside. This happens all the time in our cells. And these proteins that facilitate that, we call that the sodium potassium pump. Right? So let's say Na positive slash K positive pump. The sodium potassium pump is these proteins that pump, right, sodium out of the cell against the concentration gradient and potassium into the cell also against the concentration gradient. So by definition, active transport, this is the movement of particles, right, through the cell membrane against the concentration gradient. Against the concentration gradient. Thus, from lower to higher concentration. One example of this is the sodium-potassium pump in our cells. Another example is when plants take water through the roots, the water will move in by osmosis, but some ions present in that water, like what? Like magnesium 2 plus ions and nitrate 1 negative ions, they will be moving uh, into the root cells where they are in a greater concentration than in the surroundings of the roots. So they will be moving by active transport. So that is one example of active transport in plants, right? When plants uh, absorb mineral ions from the soil dissolved in water, the water moves into the roots by osmosis, but the ions will do it by active transport because there is a greater concentration of those ions in the roots already.